Hi, everyone. My name is Nick. I'm a PhD student in Tim Murphy's lab. And this project that I did with the CONP is called Meso-social. It's an open source approach to study mesoscale cortical circuit activity in mice during a social interaction. So we're looking at mesoscale imaging in the lab. This is imaging through the skull. We're looking at GCAMP activity. And so this is an example of what these fluorescence images look like. So we don't actually have single cell resolution with this technique, but we can make out these individual anatomical structures and the spatial patterns of activation associated with these structures. And so it's a nice non-invasive technique that we use, relatively non-invasive. So the goal of this project is to develop a simple and open source mesoscale imaging system that can be adapted to study social interactions in mice. And so to this end, we've developed a build guide with parts lists and costs for building your own Pi-based mesoscale imaging system. So this is an image of a Raspberry Pi, which is basically a small credit card sized computer. Along with this, there's step-by-step assembly instructions for completing your own Pi-based imaging rig. This is all for under $3,000. To look at social interaction, we've adapted this rig to image the brains of two mice simultaneously while they're in close proximity. And we facilitate this interaction by mounting one of these rigs onto a rail, which is controlled by a stepper motor, which is controlled by the Pi. So we can control the distance between the mice and stage an interaction between the two mice in this way. So one of the first things we noticed when we were looking at this kind of data, just looking at the average signal across both brains here, we see that as we bring the mice together, there's the activity in both of these brains tends to become more synchronized. What I mean by that is you can see these large transients occurring kind of independently when they're separate. And then when we bring it together, when we bring the mice together, we see these large events occurring more in a correlated fashion. And so we measure the Pearson correlation coefficient between these two signals. And we see that in the group data, there's a much larger correlation between the two brains when they're together than when they're in the period where they're separated before they're put together and after. And we also compared it with trial shuffled pairings. So during the together period, but with different mice. And we see there's no correlation here as well. So this kind of suggests that this correlation is more specific to the interaction than timing of events or something like that. We also looked at individual regions. So now we're looking at regions plotted on the surface of the cortex and correlating the signals between mice. And this correlation is now expressed in color on this matrix. And again, we can see a large increase almost across the board when they're together, but most notably a large increase in the posterior barrel cortex, which is the whisker-associated sensory cortex. So we're also measuring behaviors during these experiments. We've mounted a camera underneath the rigs, and we're measuring behaviors as basically as movements in these regions that we're highlighting on the video. And so these movements can look like this, and we get a movement vector which we threshold and then create a binary event vector for each of these locations. And then if we superimpose those behavior data back onto the brain data that we were just looking at, we see that a lot of these large transients actually occur coincidentally with these movements. And seeing that we saw that large correlation in the barrel cortex, looking at the whisker signal, we see that whisking initiates a large activation in the mouse brain, but it also initiates activation in the partner's brain. So moving forward, another interesting thing that we found was that it seems that the magnitude of this whisking response is dependent on the identity of the partner. So in our case, when we paired mice with cage mates versus non-cage mates, this is an average whisking response. The cage mate signal looked a lot larger. So we're hoping to investigate this in more detail using high-speed cameras to precisely track these whisking events, as well as linear models to more sophisticated method to decouple these behaviors and things like that. And we're also looking to move into more mechanistic things, looking at social motivation. So this is just a basic experiment with looking at sex preference. So this is a male mouse in the center, and you can see he's more interested in checking out the female stimulus mice. And so that's it for now. I just want to point out the UBC Brain Circuits GitHub. That's where you can find the parts list and build guide and code. Data is available on the FRDR and OSF. There's a preprint available on bioRxiv, and then this paper also has methods on the surgery. So finally, I just want to thank my colleagues, particularly Luis and Federico, and I want to thank the CONP and thank you all for listening.